Hello students, in this module I will be talking about another two methods of vegetative propagation, namely budding and micropropagation. Look, listen and learn. So let us see what is this process called as budding. Now remember, budding is a type of grafting, and how do you start it? You first basically will take the stalk and you will produce a scar on the stalk. Then you will take a bud. This time the bud won't be alone, but the bud will be accompanied by small part of the bark and living tissue. And finally you will take this bud and you will graft it in the stalk. Is it okay? And this is how the process of budding is done, which is actually a type of grafting. Let us look at a beautiful picture of budding. So right behind me you're seeing is the process of budding. Now remember, this bud has been covered with a cloth so that it doesn't fall off. But you can definitely see an extra piece of living tissue along with the bud that is basically grafted on the stalk. And the stalk is a big fat tree right behind me. Is that okay? Now remember, the process of grafting is not possible in monocots. And why would that be? Can you think of it? I think you guessed it. Monocots grafting is not possible because they lack cambium. They lack interfascicular cambium. They also lack intrafascicular cambium, and they do not really show secondary growth. For all these three reasons, grafting is not possible in monocots. So we can conclude here that because dicots show cambium, grafting is possible only in dicots. You got that right. Let us move on to the next technique called as micropropagation. Now look at this beautiful propagules of plant right behind me. So what you are seeing here now are large number of plant propagules. Is that okay? Now these large number of plant propagules are possible because of a technique which is called as tissue culture technique. Now this is another name for micropropagation. Now how do you go about doing this technique? Now for the purpose of tissue culture technique, you basically take the plant and you choose this top part which is called as shoot apical meristem. Now the shoot apical meristem can basically be used as an explant, which means you just take this shoot apical meristem off from the main plant and then use this basically to produce several copies, right? That you can call them as micropropagules. So many such shoot apices are produced and all of them are called as micropropagules because they are small piece of the plant. Is that okay? Now what do you do with these micropropagules? You then take few pots and put these micropropagules in those pots and allow these micropropagules to produce genetically identical plants, right? Something like this right behind me. Beautiful. Now what you see, all these plants pretty much look identical. Now if these plants look identical, can I call them clones? Absolutely. These plants that are genetically identical are also called as clones, right? And this technique is basically called as micropropagation, also called as tissue culture technique. The details of this technique you've already learned in a chapter called as enhancement of food production. Is that okay? So this brings us to the end of the module where I've discussed the process of budding. I've also discussed the process of micropropagation, and this brings us to the end of this module where we have discussed techniques of vegetative propagation. In the next module, I will tell you what is the main significance of the process of vegetative propagation on the whole. Till then, thank you and take care.